No, no, ya está listo.
Ya está listo. Ok, está ya. Bueno, bienvenidos. Este es el cuarto ciclo de conferencias en bioinformática y biología estructural de la Unidad de Estudios Genéticos y Forenses. Vamos a culminar en el día de hoy, 11 de diciembre del 2020, con un par de charlas, conferencias dictadas desde Pensilvania, y tenemos invitado al doctor Manuel Ospina-Giraldo. En este caso se trata de una presentación que van a hacer por partida doble. El doctor Manuel Ospina-Giraldo está en Pensilvania en este momento y Dylan J. está en New York, si no estoy mal. Dylan, correcto. Entonces, this is the last presentation of this cycle of structural biology and bioinformatics and computational biology. We have the pleasure to present and introduce my colleague and friend, Manuel Ospina-Giraldo. Is a friend 40 years ago in the University of the Valle in Colombia. Actually, Manuel is working in genomics and bioinformatics using a model, microorganism, Oomicetos, Phytophthora infestans. In this presentation, Dylan and Manuel nos to introduce your research in this area. It's a pleasure to the unit of the genetic and forensic studies and the all together to present a Manuel. Your favorite introduction, my friend.
Okay. Muchas gracias, viejo Charlie.
Okay.
Bueno, yo voy a, en primer lugar, quiero agradecerles al doctor Ramírez y a toda la gente del instituto la invitación para participar en este ciclo de conferencias. Y quiero obviamente agradecerles a todos ustedes quienes nos están siguiendo por YouTube su participación. En segundo lugar, quiero sinceramente ofrecerles mis disculpas por el hecho de que las charlas van a ser en inglés. Yo hubiera querido hacer algo distinto, pero cuando en conversaciones con el doctor Ramírez, nosotros decidimos que lo mejor era hacer las charlas en el idioma original en el que todo ha sido realizado, que es en inglés. La traducción habría sido una tarea bastante difícil, bastante compleja, y nos hubiera tomado demasiado tiempo y probablemente algo se hubiera perdido en la traducción. Así que ustedes me perdonarán, pero de todas maneras todos son bienvenidos a hacer las preguntas que consideren necesarias. Interrúmpanme si así lo necesitan. Con muchísimo gusto estaré respondiendo sus preguntas. Yo me imagino que al final de las conferencias vamos a tener una sesión de preguntas, pero si alguien necesita una aclaración adicional, con muchísimo gusto. Yo también puedo obviamente servir de intérprete o traductor para las preguntas que le vayan ustedes a realizar a Dylan. Con mucho gusto haré la traducción necesaria. Así que muchísimas gracias a todos por la atención. Entonces voy a comenzar aquí a compartir mi pantalla en un segundo. Voy a compartir la pantalla. Okay, so as Carlos mentioned, we at my lab, we work in the genomics of the causal agent of potato late blight, Phytophthora infestans. And why this organism? Well, Phytophthora infestans is a microbe, a filamentous organism that is part of a group called the Oomycetes. The Oomycetes are fungal-like organisms that for a long time have been considered part of the kingdom fungi. Way back since the description of the Oomycetes, they were considered fungi in the 1700s and so on and so forth. But since the early 1900s, I think, doubts have been generated. It became clear that Oomycetes were not fungi, that they were not part of the kingdom fungi. And there were several lines of evidence. For example, there was morphological evidence, or structural evidence, I should say. The cell wall in Oomycetes is composed of cellulose, whereas the cell wall in fungi is composed of chitin, which is the same compound that you find in the exoskeleton of insects. Okay, Oomycetes, like Phytophthora infestans, have motile stages, as you can see here. This is what we call the zoospore. It's a motile spore that has two different flagella, one short, one long. You know, the whiplash and the tinsel flagella that allows for motility for this organism. True fungi do not have any motile stages. And last but not least, with the availability of molecular studies, DNA sequencing clearly showed that Oomycetes were not fungi. And therefore, they were placed within a group of organisms that is called the chromalveolata, the chromalveolates, which are actually much more closely related to plants and amoebas than actually fungi. So here are the animals. So we are, as you can see, far from fungi as you could imagine. When we look a little bit closer into the chromalveolates, we see that the chromalveolates essentially are constituted by two groups. Now, the phylogeny of this is extremely complicated. This, by any means, has been resolved. This is a little bit like a mixed bag. And the chromalveolates are now considered part of the kingdom Chromista. This is a proposal submitted about 2 years ago by Cavalier Smith, who wrote a very good monograph suggesting why these guys should be considered part of the kingdom Chromista. With the chromalveolates with two groups, the alveolates, and this is where you see something interesting, tetrahymena, the ciliate tetrahymena, that has been very important, and an apicomplexan that I know that Dr. Ramirez will remember this, Plasmodium falciparum. These guys are actually relatively close to Phytophthora in the phylogenetic tree. And Phytophthora will be placed with brown algae and diatoms. So some of these guys are or contain photosynthetic material, and some are capable of photosynthesis. So there is some ancestor, some photosynthetic ancestor to Phytophthora, and as we will see later, there is some sort of close phylogenetic relationship with an organism like Plasmodium falciparum, which is an apicomplexan, which is the cause of the parasite that causes sickle cell anemia in humans. Now, Phytophthora has become a model for the study of Oomycetes, and that's in itself is very important. But the real importance of Phytophthora is because of what it does to plants. Phytophthora is the cause of late blight, a disease called late blight of potato, which in Spanish, at least in Colombian Spanish, is known as el tizón tardío de la papa. And this guy is characterized by the production of what we call water-soaking lesions on the leaves. So the leaves disintegrate, and obviously photosynthesis is gone, and production of tubers is gone. Now, when the tubers are actually produced, they may get infected by P. infestans, and they get rotted like this guy. These are obviously not edible anymore. There is also a historical component for the importance of P. infestans. That historical component is probably not much for us in South America, but certainly for the Irish, this is extremely important. The Irish in the 1800s, in the mid-1800s, 1843 to be exact, suffered what was called the Irish potato famine, the Big Famine, as it is called. The Big Famine actually caused the death of about 1.5 million Irish people, and the emigration of about the same number, about another 1.5 million Irish people, mostly to the United States and Canada and a few other countries in Europe. But the population in Ireland essentially was halved, so it was decreased by at least 50%. So for the Irish, late blight has a significance, a very important historical significance. Now, we're still battling late blight. There are no true good or successful mechanisms of control. And as a matter of fact, the losses, the current annual losses worldwide for late blight reach almost $7 billion. So that is just another reason why Phytophthora infestans is such an important organism to be studied. So I've been working with Phytophthora for about, I don't know, maybe 25 years, something like that. And in the last 20 or so, we decided that with the availability of new sequencing technologies, we decided that it was time to sequence the genome of P. infestans. Back then, it was still very difficult because we knew that Phytophthora was a problematic genome. There were several things in Phytophthora that made it a problematic genome. But we started doing it initially with using a whole genome shotgun approach and Sanger sequencing. And this was finished using Illumina sequence. We got what at the time was an acceptable coverage of 9-fold of about 95% of the genome. So 95% of the genome was about 230 megabases out of the 240 megabases that it is accepted as the size of the P. infestans genome. For comparison purposes, let me tell you that P. zoji, a very closely related species, and Pyramorum, another closely related species, have genomes of 95 megabases and 65 megabases, respectively. So as you can see now, right there, there is a significant difference in size between the genomes of these three different species. The number of genes that we found in Phytophthora pretty much has remained constant. You know, that number of genes usually change as annotation improves, but right now we know that it is about 18,000 protein coding genes. Now, I have to say that nowadays we do Oxford nanopore to sequence Phytophthora, and we get coverages of 35, 40x in a couple of days. This took several years to complete, so that's how things have changed in the past 10 years. But these 18,000 protein coding genes are not much different than 17,000 found in P. zoji and 14,500 or so found in Pyramorum. So as you can see, there are no changes, no significant changes in gene content. All the length is relatively similar, and there are no other significant differences, at least in what it comes to the number of genes and the type of genes. We also could not find any significant evidence of whole genome duplication or other large-scale duplication. Therefore, the question still remained: what makes Phytophthora infestans genome so large in comparison to others? Okay, once we completed the sequencing and annotation, obviously we used the very first tool that every genomicist uses, which is comparative genomics. So we compared the genome of the three closely related species of Phytophthora. And excuse me, I want to get rid of this sign here on my computer. Let's hide this. Yeah. No, that's not what I wanted. I'm sorry. Just no. I hope you're seeing the full screen as I was seeing it here. Okay. All right. Oh, okay. So we compared the genome of the 3 species, and the first thing that we found was a large core set of orthologous genes. About 8,500 genes were present either in the 2 other Phytophthora species or at least in one of them. 90% of those orthologous genes had an orthology relationship of 1 to 1 to 1, so they were present in all 3 genomes. Very interesting, the location of these genes was in conserved blocks. So we found these blocks that are represented here by these pink lines, and that indicate that the gene order is in which the gene order is exactly the same, and these purple lines that indicate that the gene order is just inverted. That's all. But what we found is that there were these blocks of genes, all of which contain a large number of genes, all of them conserved order, also with the conserved order. And very interesting, there was no indication of any repetitive DNA, so very low repetitive DNA content. Now, when we looked at what was in between these conserved blocks, we found these areas here that were really poor in terms of gene content, very few genes. But what we did find was that there was a lot of a repetitive DNA. And most of this repetitive DNA was composed of transposons, retrotransposons, and all kinds of mobile elements. And as you will see in a few minutes, this is very important for another to explain other findings that we had. So we explored a little bit more the nature of these conserved genes, and in order to do that, we decided to measure the intergenic regions at the 5' end and at the 3' end of these genes in the conserved blocks. And what did we find? Well, first, let me tell you, we did that also in P. sojii and P. ramorum. And what we found was something relatively, I guess, expected, that in most cases, or the majority of genes, had a size of intergenic region length that was more or less fixed, okay? It was, for example, at the 3' end, the intergenic regions in these guys, P. sojii and P. ramorum, were between 50 bases and about 6 kilobases or so. At the 5' end, something similar happened. The intergenic regions were between about 100 or so bases, or 150, all the way to about 3 kilobases or so, with minor differences in both genomes. Now, when we looked at the P. infestans genome, which is what we have here, what did we find? Actually, yes, the large majority of genes were concentrated here with intergenic regions that vary between 20 bases and about, 2.5, 3 kilobases or so, and 5' intergenic borders that were between 50 bases, and also same thing as about 3 kilobases or so. But that wasn't the only thing that we found. We also found that a large number of genes were located in areas where the intergenic regions were very large. All these guys here that I'm talking about, particularly this group here, you see that the intergenic regions here range between 8 and 22 kilobases on the 5' end, and similar at the 3' end. So what were these genes? Why were they located in this area? Before we can answer that question, I'll need to explain a little bit of, for those of you who may not be familiar with plant pathology, I need to explain a little bit the concept of effectors. Effectors are proteins that are secreted by pathogens and that have the effect of somehow control or modulate the plant response to the attack, to the pathogen attack. There are two kinds of effectors, apoplastic and cytoplasmic effectors. The apoplastic effectors are typically secreted by fungi, oomycetes, by nematodes also. And they are secreted into the intercellular space of the plant tissue, what we call the apoplast. These type of effectors are typically hydrolytic enzymes, toxins, and very importantly, protein inhibitors. They target very specific proteins in the apoplast, and with that, they sort of neutralize any potential defense response mounted by the plant. The cytoplasmic effectors, on the other hand, are directly translocated into the plant cell. Bacteria use what we call the type 3 secretion system that most of you probably are familiar with, and they directly target these effectors into the plant cell. Now, fungi and oomycetes use a specialized structure called the hostorium. The hostorium is a modified hypha, so a cell, a fungal cell or an oomycete cell, that is used for the exchange of nutrients. Actually, is used by the pathogen to uptake nutrients from the cytoplasm and bring it into the hostorial cell. But it is also used for the translocation or the secretion of effectors directly into the cytoplasm. Now, what do these effectors do when they reach the cytoplasm? Well, they, again, target certain kinds of proteins. Typically, the proteins that are targeted are the ones that are involved with defense response. Here, I have to say something. Not always does the pathogen succeed blocking the defense response. As a matter of fact, plants are not stupid. They have evolved over millions and millions of years a mechanism to detect these effectors. So the effectors, instead of being an attack weapon, become actually a problem for the pathogen because the plant recognizes that these effectors are present and mounts a defense response. Here's what has happened. Then the pathogen needs to evolve new effectors to circumvent the plant defense responses. This is what has been called the co-evolutionary arms race, in which pathogens develop attack weapons, plants develop resistance weapons, and that has continued for many millions of years. One kind of cytoplasmic effector that is very important is the so-called RXLR effector, which is a multi-domain protein composed of, obviously, signal peptide that facilitates the secretion of the effector from the pathogen cell. Then, after that signal peptide domain, comes another domain that has been called the RXLR domain, and probably some of you are already guessing why it is called an RXLR. It has an arginine, then any amino acid has then leucine, and then another arginine as part of this domain. It's always there. And then the third domain is what is called the effector domain. Let's focus on these two, the RXLR and the effector domain. A friend of mine, Steve Wiesen, has conducted beautiful experiments at the James Hutton Institute in Invergowrie in Dundee, Scotland. And he demonstrated that the RXLR domain is actually the one that is responsible for exclusively translocating the effector into the plant cell. So from the hostorium in the pathogen, the RXLR is the one that determines this translocation. Without the RXLR, the effector cannot reach the plant cell cytosol and remain in the hostorium, where it's useless. Now, the effector domain is very important because that is the one that actually interacts with the target inside the cell. These effector domains a lot of them have leucine zipper structures which facilitate protein-protein interactions. And that's how they interact either with the help of chaperones or directly with proteins in the cell cytoplasm. And that interaction, again, is what leads to completely modulating the resistance response and blocking it, or actually it may have the unwanted effect, at least for the pathogen, to cause a resistance response. So what we found in terms of the genome in those areas that had very low gene density but very high repetitive DNA content? Well, what we found is that about 75% of all the RXLR effectors found in P. infestans are located here. We know that our studies indicate that P. infestans has probably between 700 and 750 RXLR effectors. It's a large family. And we know for sure that about 520 are located in these areas where the repetitive DNA exists. Now, why is this important? Well, as I said earlier, these guys, the presence of transposons is going to have a recombination effect that is very significant, an increased recombination effect. This is going to be like what we call a hot spot for recombination, increasing obviously all evolutionary changes. What we believe is that the presence of these mobile elements here has somehow increased the recombination among effectors, leading to the diversification of this family at the point that Phytophthora infestans now has over 60% more effectors than any other Phytophthora. So it has plenty of attack weapons to defeat the plant. And that's why so far it is very difficult to generate, for example, transgenic plants that contain resistance genes to P. infestans. Because every time that one effector is identified as a target, a new effector actually comes into play. That's not just the only group of effectors that P. infestans has. There's also another group of effectors called crinklers. It's a little bit smaller than this, but also plays a key role in pathogenicity. But most of these are necrotrophic proteins. In other words, they actually go to kill the plant, whereas these ones just try to modulate the resistance response. So let me switch gears a little bit and tell you a little bit about a different group of genes and proteins that we think are also involved in pathogenicity, at least at the molecular level in P. infestans and potato. Although this is not just about P. infestans, now I'm going to broaden a little bit because I'm going to be talking also about research that has been done in P. solgi as a very good model for pathogenicity studies. In P. infestans, we have identified a group of genes that codes for enzymes called carbohydrate active enzymes. These are enzymes that have carbohydrates as target. There are 4 big superfamilies: carbohydrate esterases, glycoside hydrolases, glycosyl transferases, and polysaccharide lyases. Of these 4, with the exception of glycosyl transferases, the other 3 target the cell wall. So we call these guys cell wall degrading enzymes. Now, you guys have to remember something. They are targeting carbohydrates on the cell wall of the plant, like cellulose, for example. But here's the deal. They also remember that Phytophthora and Oomicetos in general have about 30% cellulose in their own cell wall. So how do they attack cellulose in the host without harming themselves? That's the question that still remains unanswered. When we scan the genome for these genes, we noticed that it was an impressive array of CASI genes in all 3 genomes, with Phytophthora obviously being the largest, 435 homologs. We wanted to find evidence about the role of these genes in pathogenicity. So one of the first things that we've done is to measure the transcriptional activity of these genes, both in mycelium, which is the vegetative state, so tissue grown in culture, in vitro, and in planta, which is the infective state, so when the pathogen has initiated infection. And for that, we used as a model, we use a group of enzymes called pectate lyases, which target the pectin, which is that cement that keeps the middle lamella together or intact, And the middle lamella, in turn, is the one that keeps the cell wall of the plant, the primary and the secondary cell wall, intact and retains, helps the cell retain its form and structure. And so we measure the activity of these genes in P. infestans when P. infestans is infecting a susceptible cultivar of soybean. Excuse me, not P. infestans, P. soji. When it is infecting a susceptible cultivar of soybean called Williams. This is a cultivated variety that does not have a resistance gene against P. soji. So we measured that, and what we found was a tremendous difference in expression between mycelial stage, so the in vitro, a non-infectious stage, and the infective stage of the pathogen. And as you can see, as time goes by, we measure this at 12, 24, 36, and 48 hours post-inoculation, or HPI. And as you can see, the vast majority of genes show or display a trend of increasing expression. This is the log2, by the way, the log2 of the level of expression. So, this is actually an exponent there. So these numbers are ridiculously high. Now, not all enzymes behave exactly the same. The upward trend is seen here, for example, in this gene 9513, increases here, and keeps increasing here. There are other genes that increase at 12 hours, like this one here, 3709, which increase a little bit here, but starts decreasing after 36 hours. We know there is probably an explanation for this. And I need to tell you this. These Phytophthora species, all of them, are what we call hemibiotrophic pathogens. In other words, they have a biotrophic stage in which they parasitize, if you will, the host, and they have a necrotrophic stage in which they actually kill the host. So at some point, the type of enzymes or genes that are expressed may vary a little bit. Initially, the idea is to destroy the cell wall, and after that, the idea is to essentially kill the plant completely. So as we see here, there was a significant increase in expression, gene expression, when we did this analysis using qPCR. But remember that in the plant is not only the cell wall, but also the cell membrane that the pathogen has to breach in order to penetrate and successfully colonize the plant. So we wanted to measure the activity of cell wall degrading enzymes versus the activity of cell membrane degrading enzymes, or the gene activity, the transcriptional activity for genes coding for these enzymes. What we found was also very interesting. In this case, we actually did this using two cultivars of soybean, Williams, which, as I mentioned earlier, is susceptible, and Williams 82, which is actually resistant, has a resistance gene for P. soji. And what we found is that actually the cell membrane degrading enzymes are expressed even at a higher transcriptional rate than the actual cell wall degrading enzymes. This was very interesting, but what was more interesting actually is how the two compare with each other. When the infection in these two cultivars starts, and I remind you, Williams is the susceptible, Williams 82 is the resistant. When the infection actually starts, most of these genes are much more highly expressed in the resistant cultivar than in the susceptible one. As time goes by, that correlation starts changing a tiny bit, all the way to here, 48 hours, when it's completely the opposite. Most genes are actually being highly expressed in Williams than in Williams 82. Why is this? Well, we understand this as the fact that the pathogen in Williams 82 has given up, has realized that there is no way it's going to win this battle, and the pathogen starts dying, and genes shut down. In this one is the opposite. The pathogen has successfully colonized the plant, is winning the battle, and the expression of these genes continues to be increased. Now, what facilitates this huge active expression, hugely active expression? Well, we think that there is always some sort of a motif in the promoter region, and we have identified at least several motifs, but in particularly one that seems to be very important in Phytophthora. This one here that we call the INR/FPR motif. INR for initiator, FPR for flanking promoter region. The initiator is this first 6-7 bases here. Then there is a spacer of 2 bases, and then from 10 to 16 is the flanking promoter region. In all Phytophthora that we have analyzed, and we have analyzed several hundred of them, in all genes that we have analyzed, we have noticed that there is somehow a variation of either one of these two components of the initiator FPR motif. Like, for example, here, this is very similar to here, and so on and so forth. Studies are underway, but it seems like this motif has a very special affinity for RNA polymerase. So much more than any other motifs that we know in eukaryotes. By the way, you guys may recall that motifs like the Tata box and the cat box are very important in eukaryotes. Well, so far we have not been able to really identify Tata boxes or cat boxes in P. infestans. So P. infestans appears to be one exception to the rule that Tata box is, the critical motif for the expression in eukaryotes. I think that's all I had to say for now. I'm sorry, I think I went a little bit over the time, but I wanted to now introduce Mr. Dylan Janchel, who has been a student in my laboratory for the past 3 years or so. And Dylan actually conducted a lot of the research that you saw on cell wall degrading enzymes. And he's now interested in has a project of his own on the study of long non-coding RNAs and the role in pathogenicity. So with that, I'd like Dylan to take over, and thank you all for your attention. I'll be glad to answer any questions at the end of this presentation. Thank you. Dylan?
Thank you. So I'm just going to open up my window. Okay, can everyone see everything okay?
Yep.
Okay. Just trying to get the laser pointer to show up.
I think you won't be able to do it until you do the presentation.
Okay, thank you. Let's see. All right, we'll just go with that.
Yeah, you can start sharing first.
Okay. So thank you for inviting myself and Dr. Ospina today. And today I'll be discussing novel long intergenic non-coding RNAs and their role in Phytophthora infestans. And these are pictures of 2 sporangia that I took a picture of while working in Dr. Ospina's lab. So let's go on. So the role of long non-coding RNAs. So long non-coding RNAs can be defined as RNA transcripts that do not encode a functional protein, and they exceed 200 nucleotides in length. And additionally, they lack an open reading frame of more than 100 amino acids. And when you compare these transcripts to mRNAs that code for protein coding genes, these are expressed at much lower levels overall. So if you see here on the right-hand side, where the central dogma of molecular biology is this typical DNA to RNA to protein, where our mRNAs fulfill this role in transcribing the protein coding gene and then being translated into protein. You can see here that on the right, long non-coding RNAs serve a different role. And these long non-coding RNAs can help regulate expression using several different mechanisms. And this is just one particular mechanism here, where it can bind to mRNAs that are mature, which may originally be used to prevent translation of a particular mRNA sequence that codes for a protein coding gene. And in this way, by sequestering these miRNAs, LNC RNAs, or long non-coding RNAs are able to toggle whether or not mature mRNAs can be translated properly. And these gene expression mechanisms can occur in both in the nucleus and the cytosol. So kind of also taking a bigger look at how these long non-coding RNAs can regulate expression in eukaryotes. You can see here in the nucleus, long non-coding RNAs can allow for different histone modifications to occur, and whether or not the chromatin condenses or expands will allow for different genes to be expressed. Additionally, long non-coding RNAs in 2 and 3 here can affect the recruitment of transcription factors and can also affect how RNA polymerase binds to the promoter of protein coding genes, which can also affect gene regulation. And also in the nucleus, you can have LNC RNAs that have a role in affecting alternative splicing, which will in turn affect which mRNAs are translated. And then post-transcriptional levels also include how LNC RNAs can bind to mature mRNAs and affect the stability of those mRNAs. And as we saw in the previous slide and here in number 6, you can see that LNC RNAs bind to microRNAs, and this sequestering effect can also have an impact on which genes are being expressed and which proteins are translated. And also, lastly, we have here 7 and 8, where you can see LNC RNAs can affect the recruitment of ribosomes, and also these LNC RNAs can, within a tissue from one cell, be transported in an extracellular vesicle to neighboring cells, where they could then affect the gene expression of neighboring cells. So specifically in Phytophthora infestans, and one of the things that Dr. Ospina has studied much in his lab is this host interaction between the host, whether it's tomato or potatoes, and Phytophthora infestans. And particularly in this paper published by Zhang et al., which is a paper that I read while working in Dr. Ospina's lab, this host LNC RNA had this role where it was acting as a decoy to sequester in microRNA. And this would affect essentially how LRR genes in the host would be expressed. And these LRR genes can recognize effectors and other plant pathogen proteins, which is particularly important for the host so that it could develop an immune response to limit colonization of Phytophthora infestans during initial stages of infection. And additionally, RNA-seq studies have been done on Phytophthora infestans, where they've looked at how metabolism changes, what pathogenesis genes are being expressed at different stages, and also like the signal transduction that's occurring. But in this paper specifically by Judelson, one of the things that I was curious about is what is the role of long non-coding RNAs? And to date, there hasn't been much exploration of this, and not many LNC RNAs have been identified. So kind of discussing the current work that's being done on non-coding RNAs in Phytophthora, and also P. infestans and P. soji. You can see here that in P. soji specifically, 940 long non-coding RNAs have been identified, and only one study has reported one non-protein coding gene family in Phytophthora infestans. And we hypothesized that using next generation sequencing data would provide high coverage needed to identify novel transcripts. And here you can see on the right-hand side, we have a picture of the Illumina sequencing machine, the NextSeq 550, which is one of the common methods that have been used to sequence transcriptomes of many organisms, whether it's Phytophthora or humans or other model organisms that are being used. So one of the key questions we asked ourselves prior to project is, how do these LNC RNAs regulate gene expression in P. infestans during asexual development and early pathogenesis? And can we identify these from transcriptome data and characterize these transcripts? So before I get into the RNA-seq data portion of it, I just kind of want to go over the asexual life cycle of P. infestans, because our RNA-seq samples that we used have been that covered basically the different life stages. So just starting off, if you can see here on the right-hand side, when you start with an infected host tissue, sporangiophores, which are essentially hyphae that have sporangia, can develop from mycelium and develop after colonization of the host. And these sporangia, if you're looking at picture C here, are essentially structures that hold spores, specifically zoospores. And these are the spores that Dr. Ospina discussed in his presentation that are motile, and they contain these two flagella. And they can act as they can initiate infection in the host. So these are eventually released from sporangia, and these zoospores, when they land on host tissue, they develop these lesions that can occur. And essentially, these zoospores, they'll develop a germ tube and an infection peg that allows them to penetrate the epidermis of the leaf, and then eventually colonization will occur of the host tissue. So going into our RNA-seq pipeline for identification of long non-coding RNAs, and specifically we wanted to look at long intergenic non-coding RNAs. So we used raw ssRNA-seq, so single-stranded RNA-seq reads that were sequenced using that Illumina NextSeq 550 system that were published by the Judason Lab, which is that paper that I had on a previous slide, where they were looking at the transcriptome changes that occurred for pathogenesis genes and other genes, either carbohydrate active enzymes or genes related to metabolism. So these raw ssRNA-seq reads were retrieved, and we downloaded them onto our server. And we filtered these reads using Trimmomatic, which allows you to filter the reads based on Phred score. And the Phred score measures essentially the quality of any of the reads and how certain the Illumina sequencing system was that particular base pair is correct at that position of the read. Next, we used the P. infestans genome and we indexed it, and reads were mapped with this, were mapped to the reference genome of Phytophthora infestans using a program called HiSat2. And the indexing step allows for the genome to be in a format that is suitable, so that way the reads can be mapped efficiently in a computational manner. And after these reads were mapped using HiSat2, the mapped reads can then be assembled with StringTie, which is, again, another program that will take your mapped reads that were mapped in the previous step and will assemble them into longer sequences and ultimately into full-length transcripts. And then once these transcripts have been assembled, you can merge all the assembled transcripts from all the different samples. And samples, some isoforms might not be expressed. Additionally, StringTie can then be used with this PrepDE Python script, which is to generate read counts for all of the transcripts that have been identified. And since we specifically wanted to look at long intergenic non-coding RNAs, we use this tool called GFF Compare, which allows you to take the GFF file that's generated from StringTie, which has the information on all the different transcripts and their genomic coordinates within the P. infestans genome, and it will use that and compare that to the known annotation file of the P. infestans genome in order to determine whether or not a transcript is overlapping with known exons, whether or not it's intergenic, whether or not it's antisense, meaning that the sequence is moving in the opposite direction of a known coding gene. So specifically, we wanted to look at class U, which is defined as these intergenic transcripts that do not overlap with previously known annotated genes, whether or not these are protein coding genes or rRNAs or tRNAs. Specifically, we wanted to see these intergenic sequences. And these range between at least a 2,000 base pair difference or distance between any known annotated sequences. So this list of class U transcripts were then filtered to make sure that they had more than 200 nucleotides in length, and that also each transcript had less than 100 amino acids in their open reading frame. And then we were able to calculate the coding potential using a program called Coding Potential Calculator 2. And then from this list, we selected transcripts with less than 0.2 coding potential. And then these counts for the different, the read count specifically for these transcripts were then loaded into R, and log CPM were generated using EdgeR. So the EdgeR pipeline for kind of going through, once these read counts are loaded, you could then use this to generate log CPM matrices of the different transcripts across the different samples. So first, when we loaded these into R, we wanted to remove any transcripts that were expressed so low that their results would be hard to interpret in the log CPM value, and this makes sure that these transcripts are actually real transcripts that are being transcribed among the different samples. So it goes across all the samples to make sure that across all samples, each transcript had a minimum of 15 reads across the different samples that we're using. And once we did this step, we found that there were 872 unique long intergenic non-coding RNAs and 187 isoforms that were identified. And once we did this expression filter step to make sure that all the long non-coding RNAs that were very lowly expressed were removed, we normalized the library sizes using TMM normalization, which is a common step used in this EdgeR pipeline. And it essentially just makes sure that if you have few genes within a sample that are being expressed highly, that could allow people to misinterpret whether or not other genes that haven't changed expression are being downregulated. So this library size normalization step is particularly important. And after that normalization step, we were able to generate principal component analysis plots of the samples to see how the replicates were clustered together, and then also how the larger sample groups were clustered together. And then log CPM values were calculated for all the replicates. And across the different replicates, we averaged each group, so that way we can do comparisons among either mycelium group or a sporangia group or other pathogenesis stages. And then it allows us to generate a heat map of the top 50 variably expressed long intergenic non-coding RNAs. So going through the length and GC content of these transcripts and kind of comparing them to what we see in PSOG and also just the GC content in genomes of Phytophthora species, we see here that the length and GC content was 615 base pairs or 539 base pairs for the mean and median respectively. And the GC content was around 48%, whether it was 48.3 or 47.9 based on the mean or median. And we have here box plots on the right-hand side where you can visualize what the length of the long intergenic non-coding RNAs were, and also histograms to kind of show you the distribution of these values. So looking at the long intergenic non-coding RNA histogram, you can see that most of these transcripts are less than 1,000 base pairs. And even so, most of them occupy the bucket of either being less than 250 or less than 500 in this first two columns here. And when we compared the reported PSOG length and GC content, the median GC length of these transcripts were 558, and whereas ours was 539, which we were happy to see because this was pretty close. And then GC content in Phytophthora genomes, P. infestans is known to have a lower GC content compared to PSOG. So it was kind of expected that we'd see that the long intergenic non-coding RNAs that we found here have a much lower GC content than what was reported in PSOG. And then looking at our clustering of our samples, so there was 5 different life stages that we were analyzing from this data set, and then also 6 different infection states. So our mycelium group, which is our in vitro mycelium of the pathogen clustered together. And then we have zoospores, which are these motile spores, which didn't cluster together as well, but these are something similar that we saw in the Judelson's paper where they didn't cluster together among protein coding genes as much. But other than the zoospores, germinating cysts, chilled sporangia, or sporangia clustered together quite nicely. And in this red box, you can see here all of these infection states were all grouped together. And if we zoom in on this and look at how these are clustered, we can see that by color code, whether it was potato leaflets at 1 day post-infection, or tomato leaflets at 1 or 2 days post-infection, or tubers of potatoes are 1 or 2 days post-infection, we can see that they cluster together. And then looking at our heat map that we were able to generate of these different transcripts, we found four groups that we'd want to further investigate. Specifically, this first black box here on the top right-hand side of the heat map, you can see these are transcripts that are enriched in either potato leaflets at 2 days post-infection, or tubers 2 days post-infection, and tomato leaflets 2 days, as well as tomato tubers and potato leaflets at 1 day post-infection. And when you compare that to mycelium for that same row, you can see that these are not enriched in mycelium. So these are likely long intergenic non-coding RNAs that are being enriched in these stages that could allow for modulation of gene expression of different pathogenesis genes, whether it's RLXLR effectors or carbohydrate active enzymes. And we see among our other 3 boxes here, our second box, this is a second cluster of genes that could be highly expressed in that are enriched in these particular stages, potato leaflets, tea leaflets, or tubers. And these rows, or the color of these cells within each of these transcripts, are based on essentially a z-score, where it uses for each particular transcript, the z-score is scaled based on the log CPM of that transcript within each of the different samples. And the average among all the samples is taken, and the standard deviation is taken in order to get a z-score, which will then be reflected in the color of these heat maps. So in our third and our fourth plot, you can also see that we have a cluster that is really enriched in zoospores, germinating cysts, and chilled sporangia, and a cluster that's enriched in mycelium as well. And kind of our outstanding questions that we want to further investigate is, what are the binding targets of these transcripts? Are they binding miRNAs, or are they binding mature RNA transcripts? And is the expression of these long intergenic non-coding RNAs correlated with the expression of RLXLR effectors and carbohydrate active enzymes? And specifically, like, is it at 1 day post-infection or 2 days post-infection? Which is particularly important because at around 1 to 2 days is when you start to see the shift from biotrophy to necrotrophy, which is particularly important for Phytophthora infestans. And then we also would like to see how these 4 groups are clustered together, or if they're dispersed throughout the Phytophthora infestans genome, and whether or not these long intergenic non-coding RNAs are located in gene-rich or gene-sparse regions. And I just want to say thank you to Dr. Ospina and Dr. Ho for helping me with this project, and for the staff at Lafayette College that have helped with the lab and making sure we have the supplies we need, and making sure the server is running well. And Thank you to Dr. Ramirez for inviting me for this presentation. And thank you for listening.
Yes, Dylan, thank you. Thank you for all of you. This is a question, Rafael, in the chat. Well, I have a question to what?
Mm-hmm.
Is RNA so important to the expression of genes, effectively? What are the proposed biological proposed for this sequence in the sense of the evolution part or the genomic part?
Oh, and what is the role of like long non-coding RNAs in evolution? Is that kind of what you're asking, or?
Yes, exactly.
Yes. Yeah. So specifically, the main role is making sure that gene expression is being regulated and properly. So if we're looking at in plant pathogens specifically, it's to make sure that if we're looking in Phytophthora in that PSOG paper that I had mentioned, they found that these transcripts are correlated with the expression of pathogenesis genes, whether it's like effectors. If we're looking in humans per se for long non-coding RNAs, they found that a lot of them are involved in tissue-specific differences. So in comparing tissue in like the liver compared to tissue in immune cells, like these long non-coding RNAs have that particular role, and they're less conserved among between species and between species, and then also between different tissue types. So this greater like variation allows for which proteins to be expressed in either different tissues to be more tuned properly, if that makes sense.
Yeah. Thank you, Dylan. To Manuel.
Mm-hmm. ¿Qué pasa?
In Spanish.
Sí, está bien, en Spanish. Sí.
Ok. Manuel, recordando nuestras andanzas en el área de la botánica hace algunas décadas atrás, siempre recuerdo mucho la parte de la sistemática, ¿no? Y hoy día tenemos herramientas moleculares muy importantes para hacer la sistemática, pero no solamente para eso, sino para entender un poco biológicamente qué está sucediendo con estos organismos, ¿no? Y la pregunta para Dylan tenía que ver un poco con si los genomas evolucionan y coevolucionan específicamente, ¿cómo es esa relación huésped-hospedero en el caso de las solanáceas, papa, tomate y este tipo de cosas? Tiene que ver mucho con cómo se han manejado esos cultivos a través de 10.000 o 15.000 años de civilización y de cultura, ¿no? ¿Qué opinión tienes al respecto sobre ese aspecto, Manuel?
A ver, una de las cosas, puedo responder en español, ¿cierto? Creo que sí.
Yes. Sí.
Una de las cosas que hay que tener en cuenta y que ha sucedido mucho en el campo de los oomicetos es el hecho de la recombinación sexual ha tenido un efecto muy grande en la eficiencia en la diversificación de todas estas especies, primero que todo. Entonces, ahora, el hecho de que ha habido monocultivo por tantos años, como vos lo sabés, como ha habido tanto monocultivo, digamos, en lo que se relaciona con papa, banano, café, todos esos. Todo eso ha facilitado que patógenos hayan tenido una posibilidad muy grande de adaptación. En el momento, actualmente, por ejemplo, tenemos el problema con banano, con café, que un patógeno, uno solo, puede borrar completamente todos los cultivos. Entonces, de monocultures en general ha sido uno de los grandes problemas que ha habido en fitopatología, porque eso ha facilitado la evolución de los patógenos bastante. Yo creo que de pronto otros aspectos que hay, vos mencionabas lo de las herramientas moleculares, y lo que vemos es que la evolución ha sido tal que ha generado casi siempre una respuesta constante de patógenos y de huéspedes y hospederos. Una respuesta constante. De eso hay siempre una respuesta constante. El hospedero desarrolla una respuesta de defensa y el huésped entonces desarrolla una respuesta de ataque. ¿Qué ha sucedido? Yo creo que no todos los problemas lo podemos ver dentro de los últimos, desde que la agricultura fue domesticada, desde los egipcios empezaron a domesticar agricultura hace 10.000 años. Yo creo que incluso desde antes tiene que haber ha habido una constante renovación. Ahora, hay una cosa interesante con Phytophthora, particularmente en México. En México, que es considerado el centro de origen del patógeno. Ambos patógeno y planta han estado evolucionando por muchísimos años y de hecho es en México en donde encontramos más resistencia al patógeno. Generalmente resistencia que ha sido integrada de especies que son especies silvestres. Esa resistencia ha sido integrada en especies cultivadas. Y ha tenido en México, ha habido a pesar de que en México hay recombinación sexual del patógeno, la ha habido siempre y hay una gran diversidad genética. A pesar de ello, en México nunca ha habido el efecto, por ejemplo, del tizón de la papa no ha sido tan intenso como ha sido en otros lugares. Ahora, por ejemplo, aquí Manuel, en Estados Unidos...
Sí, por ahí hay una pregunta, disculpa, que está un poco relacionado con lo que estamos compartiendo. Y tiene que ver, es una pregunta que está haciendo uno de nuestros estudiantes aquí de agroecología, específicamente, asociado con el uso de los fungicidas, ¿no? Hay una presión, evidentemente, sobre esa especie en particular.
Sí.
Y me llama la atención lo que acabas de decir en cuanto a México. Es el origen de esto, pero también tiene diversidad. Entonces la diversidad genética muy es grande. Importante, muy importante, pero también como un mecanismo de respuesta ante incluso la presión selectiva que se está dando utilizando los agrotóxicos, ¿no?
Sí, sí, en eso hay una cosa muy importante que decir. La presión selectiva de fungicidas ha sido muy grande y, por ejemplo, aquí en Estados Unidos, hasta 1990 o algo así, algunos fungicidas fueron bastante exitosos. En los 90, 91, 92, Phytophthora es un organismo que se reproduce asexualmente, pero también se reproduce sexualmente, a diferencia de muchos otros. Tiene un efecto muy grande cuando se reproduce sexualmente. Necesita two mating types. Necesita dos tipos, el A1 y el A2. Okay. Aquí en Estados Unidos solamente había uno hasta 1990, creo que era el A1. Y en 1991-92 llegó el A2. Y la recombinación sexual empezó a darse en todas partes. ¿Qué pasó? Lo primero que sucedió fue que Phytophthora empezó a mostrar resistencia contra una fungicida que se llamaba Venomil. Y ya ese realmente no es funcional para controlar Phytophthora. Entonces, eso es otro aspecto. A pesar de que hay alguna diversidad aquí, esa diversidad es relativa. porque a la mayoría de esa diversidad viene de variedades cultivadas, y no es diversidad
No son
natural.
naturales.
No es diversidad natural, como lo es en México o como lo es en otra zona donde también ha sobrevivido patógeno y planta han coexistido, en Perú. En Perú y más que todo en Perú ha habido una relativa coexistencia de patógeno y planta, porque la presión de selección contra el patógeno es grande, pero aún así el patógeno tiene algo así como su propio nicho, en donde esencialmente ha sobrevivido sin problema. Hay pérdidas, obviamente, se pierde mucha la papa por el tizón. Y en Colombia creo que también el problema del tizón de la papa todavía existe, pero hay más diversidad y esa diversidad es importantísima, por lo menos para el mantenimiento de un hospedero válido, pues, de un hospedero que sea resistente o que no tenga mayores pérdidas, eso que es lo más importante.
Tengo otra pregunta desde el punto de vista técnico, ¿no? El utilizar todas estas herramientas moleculares, pues nos ha permitido conocer mucho más sobre eso, los genomas en particular. ¿Tú crees que haciendo todos estos estudios comparativos a nivel de transcriptómica, nosotros podamos tener, digamos, a un tiempo prudencial todo el conocimiento necesario para hacer mejores controles sobre estas plagas en particular? Me preocupa porque son plagas de alimentos esenciales para la humanidad, ¿no?
Sí.
Y eso es fundamental, ¿no? Pero vamos muy rápido en los últimos, tal vez 20, 30 años, cuando recién iniciamos los estudios de genética. Tú recuerdas que se sabía muy poco sobre estas cosas. Estamos muy rápidos hoy día y cuesta un poco entender a veces para los estudiantes, en particular los que están iniciando su carrera, entender que las herramientas moleculares nos ayudan un poco. Pero los análisis, sobre todo los análisis que podamos obtener, porque hay mucho dato y los análisis bioinformáticos son muy importantes, son elementos de valor para el conocimiento, ¿no? No solamente es el trabajo en el laboratorio, sino después cuando vienen los datos, y esos datos pues son fundamentales. ¿Cómo tratar ese tipo de cosas y cómo debe un profesional con el nuevo reto de estas herramientas prepararse para ese futuro que es a corto plazo? Ya casi.
Bueno, hay varias partes en la pregunta tuya. La primera es si con el uso de estas herramientas vamos a tener un éxito a un futuro plazo, digamos, cercano. Hombre, yo no quiero sonar demasiado pesimista, pero yo veo que es muy complejo el combatir estos organismos que realmente tienen una actividad biológica ilimitada. ¿Sí? Si pensamos, por ejemplo, si comparamos un vertebrado, un humano, por ejemplo, nosotros tenemos una actividad biológica bastante limitada. Reproducción sexual, que tiene que estar dentro de ciertas normas y así por el estilo. Estos organismos no tienen ninguna barrera en lo que se refiere a reproducción. Las posibilidades evolutivas en estos organismos es infinita. Entonces, en eso nos llevan una ventaja impresionante. Por supuesto que todos estos estudios transcriptómicos, Dylan said something about the study from Howard Jodelson, is a guy who has studied the biology of bee infestans for a long time and has provided, very good knowledge. Entonces, el trabajo de Howard Jodelson es muy importante, extremadamente importante. Pero yo creo que encontrar, digamos, una cura o una forma de combatir el patógeno de manera definitiva, no creo que va a ser fácil. Yo creo que lo único que nos puede mantener un poquito a nivel con el patógeno es la diversidad. La diversidad biológica va a ser lo único. Eso me lleva a la segunda parte de tu pregunta. Por ejemplo, bueno, en lo que presentamos aquí fue una parte muy pequeñita de lo que hemos hecho, pero llevamos, yo soy, si se quiere, de la gente más nueva en fitopatología de Phytophthora, y yo llevo 25 años trabajando en eso. Hay mucha gente que lleva Bill Fry, Jodelson, Brett, Tyler, esa gente lleva 35, 40 años trabajando en esto, y aún así saben, y ellos mismos lo han dicho, cada vez que vamos a conferencias ellos mismos han dicho, la situación es compleja. Sabemos mucho más, pero cuanto más sabemos, más nos enteramos de lo que no sabemos.
Exacto.
Cuanto más sabemos, descubrimos que hay tanta cosa que no sabemos.
Bueno, yo creo que ese es el elemento clave en el desarrollo del conocimiento.
Ese es, y las nuevas generaciones probablemente tendrán que entender que en el laboratorio uno trabaja constantemente, pero ese gran descubrimiento, eso no llega de la noche a la mañana.
Okay.
Eso no llega de la noche a la mañana. Ahora, la bioinformática para nosotros ha sido una bendición. Es decir, la bioinformática realmente explotó a partir de 1995 de pronto, o a partir del año 2000 como tal. Se ha explotado pues como ciencia. Entonces las nuevas generaciones tienen esa ventaja. Ellos, como le digo yo a mis hijos, ellos nacieron en la época de la informática. Nosotros no. Nosotros no. A nosotros nos tocó hacer cositas una por una. Secuenciar con Sanger, hacerlo y medir las bases una por una.
Autorradiografía.
Nada de, por ejemplo, en mi laboratorio, Dylan y otro estudiante, Nick Grams, ellos hicieron la secuenciación usando Oxford Nanopore y obtienen millones y millones de data en 2 días. Eso no era así. No. Entonces, pero de todas maneras, el hecho de que uno pueda hallar ese conocimiento ahora más rápido, eso no, de todas maneras no garantiza que vayamos a responder todas las preguntas inmediatamente. Y las nuevas generaciones probablemente tendrán que ser pacientes, aplicar bioinformática. Y hay otra cosa importante, y que es algo que conversábamos el otro día, la necesidad de asociar la biología con la informática. Informática sin biología, creo que no tiene sentido. Hay que saber la biología del organismo, saberla en profundidad.
Claro.
En la biología del organismo, poca informática podemos usar.
Eso es importante. Rafael tiene un comentario.
Thanks, Dr. Manuel and Dr. Dylan for amazing presentation. I have maybe a question more technical or more for curiosity about bioinformatics. How is the process? But I working with genome assembly of bacteria, no? If some issues or some concern to do this in the correct manner. How is the complex assembly of genomes and the Phytophthora? How maybe the polyploid ploidies? How you can handle this problem, this work?
So I think the question might be more directed to Dr. Ospina, just because I think that's like specific about the genomic sequencing. I've specifically been working on like the transcriptome sequencing, which you don't have to worry about that because you're sequencing RNA. So I'm not sure if Dr. Ospina wants to answer that, but in terms of like an RNA-seq perspective, once you have the reference annotation and the genome sequence, it's a matter of mapping those reads onto the known sequence and then assembling them together, which can be used using like a variety of tools. The ones that I happen to use were HiSat2 and StringTie, which are 2 programs that have been published in Nature Protocols paper in like 2016. So I found those were like pretty recent tools that you could use to assemble these and map and assemble the reads.
Uh-huh. Yo puedo agregar que el ensamblaje de todo el genoma, bueno, también depende de qué estemos hablando. En el caso de Phytophthora, el organismo es diploide.
Diploide.
Que eso pues hace las cosas más complicadas. Esa es otra de las diferencias con la mayoría de los hongos. La mayoría de los hongos son, en la mayor parte de la vida, el ciclo de vida es asexual, they are haploide. Mientras que Phytophthora pasa la mayoría de la vida como organismo diploide. La parte de haploide de Phytophthora es insignificante. Es insignificante. Es lo que cuando se produce una cosa que se llama el oogonium y el antheridium, que es como en plantas, ahí se usan términos similares. Entonces, primero que todo, ese es uno de los grandes problemas. Pero yo creo que en los últimos tiempos hay muchas herramientas que son fácilmente accesibles. Por ejemplo, Dylan, correct me if I'm wrong, but everything you use was available on the internet? ¿Sí?
Yes. So you can use your born again shell, if you're using a Mac, you can access that in terminal. And you can, depending on like your organism, the tools you can just download and install on your own computer, and they have like the documentation available. So like I didn't have to pay for like any of the tools. But in terms of like data sets, like if you're studying a particular organism and there's other groups that have like published RNA-seq data sets, or you want to do an RNA-seq on your samples, you could then download like the tools in order to do the mapping for the reads, assuming that you've already have like a genome sequence that has already been like published and annotated.
Y si tienes, por ejemplo, bueno, cosas que siempre vas a necesitar es una buen soporte computacional. Computadores con procesadores que sean capaces de manejar la cantidad de información, porque estamos hablando de, por ejemplo, en el caso, en la última secuenciación que hicimos de Phytophthora con Oxford, creo que yo creo que tengo ahí en el laboratorio, creo que solamente en ese experimento, más de como 450, 500 gigabytes de información. Entonces, ese es otro problema. Pero, por ejemplo, Arachne, que se usa para ensamblaje, se puede conseguir.
Justamente, Manuel, disculpa, ahí interrumpo un poco porque hay una pregunta desde Perú. Joel Torres nos pregunta específicamente de qué manera o con qué software en particular puedo mapear RNA no codificantes en genomas de referencia, en el caso de Phytophthora o cualquier otro de estos patógenos, ¿no? La pregunta está asociada con si esos genomas de referencia, ya sean RNA monocatenario, se pueden utilizar algún programa disponible. Siempre tratamos, uno de los problemas nuestros, lo conoce de América Latina, el acceso a recursos informáticos, y entonces si hay programas disponibles y cuál recomendarías para trabajar en este caso, o con cuáles tienen experiencia ustedes en particular.
Para RNA, la mayor parte de los programas que usamos en el laboratorio, bueno, son los programas que mencionó Dylan. Creo que
Dylan, yes.
Dylan, really for RNA assembly and for transcriptome assembly, I think the programs that you use are the only ones that we have used in the lab. Is that right, that you have used?
That is correct.
Yeah. So I guess lo importante sería, como decía Dylan, la mayoría de esos programas se pueden bajar, se pueden descargar y instalar en una estación en una estación local. Entonces, Dylan, for your assembly, you used initially, what was it again? You went through HiSat and...
HiSat is to map the reads, and then string-tie will take the mapped reads and will assemble those into transcripts.
So those two would be, esos dos serían los más, probably los programas más utilizados que podrían usar. Yo he oído mucho de HiSat y de string-tie. Esos dos programas los he oído mencionar y la gente los usa bastante. Y pues esa fue la razón por la cual los empezamos a usar en el laboratorio. La mayoría de la gente con la que yo trabajo y que ha hecho este tipo de estudios ha utilizado esos. También, Dylan, I don't know if you came across top hat and cufflinks.
Yes, I came across both of those. And I know HiSat is like one of the newer ones that is used to do the mapping.
Mm-hmm.
And then regarding string tie, I think one of the benefits of string tie is like being able to identify the novel isoforms, which some of the other assemblers don't do as well.
Mm-hmm.
But yes, like I've come across both of those, and I ended up deciding to use the HiSat string tie pipeline, which that string tie tool has been developed by like faculty at Johns Hopkins, like computational biology. So like, and like that paper has been cited like many times, and it was pretty recent. It was in 2016. So that's the pipeline that I ended up trying to use, and then import that into EdgeR, which is how the Judelson lab ended up doing the actual like expression analysis to see like, you know, the log CPM of the different genes.
Manuel, you have a core facility in bioinformatics in the university?
No, we don't. Well, we have an IT department that helps a lot, but we don't have a core facility. We have in our department, we do have a bioinformaticist. It's a guy who we have collaborated a lot, and he has relatively good servers. So we don't have. But person in Peru, Joel, que estaba preguntando, yo me acuerdo que Penn State ofrecía un servicio que se llamaba Galaxy, en donde había muchas de estas herramientas estaban disponibles. Now, ellos todavía usaban Top Hat y Cufflinks, pero como dijo Dylan, HiSat es un programa mucho más nuevo y que ha demostrado pues que tiene, es mejor en materia para todo el ensamblaje.
Más eficiente en el mapeo.
Más eficiente que los que conocíamos antes.
Una last question about this genome assembly. You use two approach to technology to sequencing, no? Maybe nanopore and Illumina.
No, para la primera publicación que hicimos en Nature del genoma, para esa época estábamos terminando con Illumina. Se terminó la mayoría con Illumina y se comenzó con Sanger. Nosotros todo lo habíamos comenzado con Sanger.
We have MySQL in this unit.
Okay.
And also Sanger. But it's very expensive to also new technology.
Very expensive, but in Phytophthora, mostly because of the repeats and because of we knew this is something that didn't come as a surprise. We knew that Phytophthora genome was very difficult. We always say that, you know, and we were talking about this the other day, DNA is all the same. No, it's the same, no. Yes, but genomes are not.
My question is this: with you have a mixing approach, sequencing both Illumina and then a nanopore to close the gaps around the genomes. You use it's more useful for difficult genomes. Like you agreed with this approach. You use this approach with the sequencing in both sequencing with Illumina
cortó cuando se cortó la no oí lo que me, hay una parte que no me dijiste, que no te entendí porque se cortó la comunicación, se congeló. La pregunta era que si los dos, que usar los dos métodos o diferentes métodos. Mira.
Sí, mejora el ensamblaje, porque bueno, con Illumina es más pequeño.
Muchos gaps.
Y con nanopore pueden cerrar esos gaps, ¿no? Para
sí.
Phytophthora igual aplica eso, ¿lo han realizado?
Mira, se ha hecho solamente con Illumina, pero necesita un poder computacional bastante grande, porque vos sabés que con Illumina lo que conseguí son 300 bases.
Pequeños,
250.
150 en el mejor de los casos, si es en un unidireccional, 300 si lo haces en las dos. Entonces, eso realmente requiere un poder computacional muy grande. Vas a terminar con una cantidad de fragmentos increíble. La ventaja de Nanopore es que con Nanopore, si todo funciona bien, vos conseguís reads que son fácilmente 10.000, 12.000 bases. Entonces, un config puede tener varios millones de bases y rápidamente el ensamblaje es muchísimo más rápido.
Más rápido y más eficiente.
Más eficiente. La confiabilidad es buena, pero hay que decirlo, Sanger todavía tiene ciertas ventajas en algunas secuencias.
Gracias por haber hecho esa última aclaración. Es importante para los estudiantes aprender de esa técnica y saber la potencialidad que tienen, aun cuando tecnológicamente pareciera que están un poco atrás en la nueva tecnología. No se superan todavía.
No puede usar un Ferrari, para hacer cross country, una cosa así.
No, para nada.
Una vaina.
Excelente.
Exactamente.
Excelente. ¿Tenemos más preguntas por acá? Por ahora no tenemos más preguntas. Se han conectado en reproducciones. Estamos viendo las estadísticas aquí. Hay 47 personas y simultáneamente en este momento hay 12 personas simultáneamente. Tenemos una lista de personas que se inscribieron. Creo que te lo dijo Rafael desde el principio. No estamos en línea en este momento. Más de 70 personas que seguramente están interesados. Y la ventaja de poderlo dejar en YouTube colgado es para intercambiar esa información. Así que, por favor, denos su contacto si quieren compartir.
Por supuesto. Alguien que tenga preguntas o que quiera saber más al respecto nos puede contactar. Mi correo electrónico es ospinam, Ospina M, más fácil así, Ospina M, @Lafayette, LAFAY.
You spell it difficult for this moment in Spanish or English?
Eso yo lo puedo pronunciar más en inglés que en español.
Your Turkish is better than
eso es mejor.
This is difficult.
Ospinam@lafayette.edu. EE.UU. Y yo te puedo mandar ese mensaje también.
Dylan, ya lo acabamos ya en el chat del grupo lo acabamos de colocar.
Pues claro, ahí, espérate un momentico, yo puedo, espérate, te pongo mi dirección y después
pongo
Dylan, y después pone Dylan.
Your email address, questions.
Yes, so I can give you my Gmail account. It's DYLAN, Jane, so just my name, DylanJaneshill@gmail.com.
Yes.
All right.
So that's...
All right.
Igual toda esta información y el y las conferencias van a estar grabadas en nuestra página web. Y pueden acceder desde allí también y preguntar y volver a ver
dejar
la
comentarios.
conferencia, dejar los
listo.
comentarios.
Claro, y yo lo puedo, yo me
las
imagino que recibo la
compartimos por acá.
notificación.
Exactamente.
Ok.
Se las hacemos llegar. Entonces, bueno, creo que ha sido una gran conferencia y un gran intercambio científico.
De ideas.
Y bueno, la posibilidad de que nos regalen su tiempo para poder conversar de esta forma amena y de compartir y divulgar el conocimiento, que al final es lo objetivo de este ciclo de conferencias. Muchas gracias por esta clausura.
A ustedes muchas gracias por la invitación. Ha sido una oportunidad muy grande para presentar lo que hemos hecho y sobre todo para reconectar y esto de los que es muy importante.
Y bueno, a ver si el año que viene lo comprometemos a ver si podemos organizar un pequeño curso.
Un curso about this problem. It's important to us, important to all. Effectively.
Absolutely.
Yes.
Yeah, absolutely.
Many thank you for both. This is opportunity to exchange information and for to see you and again after all these years. Yes. You need to hear the Beatles, Rolling Stone, and to make the broadcasting about the Beatles. You remember?
Yeah, should do that too.
Yes.
Okay. All right, guys.
Bueno, pendiente. Muchas gracias.
Dylan, thank you for being available too. So thank you very much. We'll be in touch.
Thank you for inviting me today.
All right. Thank you, guys.
Bye.
See ya.